let's talk about endocrine anatomy and physiology here. Okay, it's an important concept. Many hormones are involved in the regulation of a number of body functions, and we need to know which hormone does what and when and how, those kinds of things. And it's very detailed, and therefore this would be a set of recordings, not just one recording. Okay, the first one is looking at the basic characteristics of hormones, the definitions, and details like classification of hormones. Okay, so let's get ready. Before we even get to the hormones, let's talk about the key ways by which chemical substances bring about their effects at the target cells. Autocrine, paracrine, endocrine, and neuroendocrine means by which chemical substances act on the target cells. Let's understand each type here. Autocrine signaling, look at the name, auto means self. It's acting on itself is what you get out of this term. Now, the illustration here shows a cell with a vesicle that's filled with a chemical substance, a secretory vesicle, right? So now this chemical is released by the cell, and that's shown here. And this chemical then binds to its receptor on the same cell. And then perhaps it makes the cell release more chemicals, and that would bind to the receptor on the cell and trigger this response over and over. Typically, though, in the body, this kind of example is common in the immune response. Helper T cells are a good example here. Cytokines released by helper T cells would act like this. They bind to the helper T cell, trigger the helper T cell to release more cytokines and more cytokines. Okay, now let's talk about the paracrine signaling. Para means around. Okay, so the cell that makes a chemical substance, like in this example, would release its chemical, which then acts on a substance nearby. Which acts on a cell nearby. So here is one cell. Let's say this is the cell right within the vicinity. So the chemical that is released by this cell will travel just a short distance until it reaches its target cell receptor somewhere close by. Okay, a good example of this type of communication is prostaglandins. Prostaglandins were given the name because they were first discovered in the prostate gland. However, these local hormones happen to be made elsewhere too. Endothelial cells make prostaglandins, platelets make prostaglandins. So the name does not apply to prostate gland only anymore, but the original name has been retained. Anyway, what we learned here is there are local hormones. They are released within a tissue, and they have their target cells within the same tissue. They do not have to travel too far to bring about their effects at the cell. Now let's get to the actual detail of hormones. Okay, this is endocrine signaling. Endocrine signaling is where we talk about endocrine gland, hormone, target cell, its transportation, a lot of things in one slide. Okay, so what you're looking at here is an example of an endocrine cell. A hormone is made, stored here, released. When it's released, it's getting into the blood. Okay, and that's classic of endocrine gland. Okay, why? Because endocrine gland does not have a duct of its own. Okay, so now we'll take a step backwards to talk about what this duct is all about. and where it comes from, etc. So let's look at how glands are made. Epithelial cells are the ones that form glands. Epithelial tissue forms glands, correct? Now, the epithelial tissue has a characteristic feature that it doesn't have much space between the cells, okay? Limited extracellular space. So the gland that's made is typically placed in the underneath connective tissue. Fine. Some glands have their own ducts, so when they make a substance such as saliva or sweat, it goes through the duct and then is released on the surface of the gland. Okay? So that is exocrine gland. Now, what happens to some glands is that once they're formed, there is no duct that's retained. Those are endocrine glands, ductless glands. So there is no duct to release the substance. The substance is released simply into the extracellular space. And there happens to be a special type of capillaries. These are called fenestrated capillaries. Porous nature of these capillaries allow the passage of hormones into a bloodstream. So that's the example you see. Hormone is in the blood, and hormone can be transported throughout the body via blood. However, when the hormone finds its receptor on a target cell, it leaves the blood and then binds to its receptor, and then bring about a specific change within the target cell. So that is how the endocrine gland works. It releases a substance, it's called a hormone, and travels in the blood until it finds its target cell, and then leave the blood to bind to its receptor at the target cell. Okay, now the next one is basically the endocrine function, but carried out by a neuron. Neuron is an endocrine cell here. That's all there is. Okay, so this is a neuron. It releases a hormone. Okay, you could call this as a neurohormone if you want it. Okay, so that again shows the same behavior we have talked about. Reaches the blood, travels in the blood until a target cell receptor is found. Okay, a few examples of neurons serving as endocrine gland would be hypothalamus and anterior pituitary gland. We will look at those in the later part of the chapter. Okay, with this, let's move on to talk about hormone transportation. Well, it all depends on the nature of the hormone, which chemical substance is this hormone made of, is how the transportation is determined. Okay, some hormones are actually, majority of the hormones are water-soluble hormones. They go in the solution, and that's how they travel in the blood. Whereas some hormones are not water-soluble. Okay, they are steroid hormones and thyroid hormones. Thyroid hormones T3 and T4. Steroid hormones, you can think about estrogens, progesterone, testosterone, cortisol, aldosterone. All those are steroid hormones. They do not dissolve in the blood, and therefore they need carrier proteins. Carrier proteins are nothing but plasma proteins. So the advantage of hormones being bound to carrier proteins is that they would have a longer half-life. Okay, they are degraded less readily when they're bound to carrier proteins than when they're freely available in the blood. Okay, so that's how they have longer half-life. Okay, now a quick comparison of hormones to the quick comparison of the endocrine system to that of the nervous system. Okay, both these systems are regulatory systems, control many functions. And how are they similar, how are they different is what we really want to get out of this, okay? So signaling mechanism in the endocrine system is always going to be via hormones, which are chemical substances, whereas in the nervous system you have neurotransmitters, those are chemicals, and then the electrical signal, that is the action potential signal they use. Then the primary chemical signal in the endocrine system obviously is hormones, whereas the primary chemical signal in the nervous system are neurotransmitters. Distance traveled is about the same, long or short for endocrine system. Remember, they could travel in the blood throughout the body, and or they could have an endocrine gland very next to them, like hypothalamus and pituitary. In any case, in the nervous system, the distance traveled could also be long or short. For neurotransmitters, it's not a long travel typically. It's usually presynaptic, postsynaptic cell in a synapse, and there is very little space between the two cells, and the neurotransmitter released would bind to the receptor at the postsynaptic cell, and that's how long the distance is. Some neurotransmitters, when epinephrine and norepinephrine are released into blood, that is when they do travel a bit. And action potentials travel short or long distances. You could think about an action potential generated at the very tip of the foot, and that has to travel all the way along the neuron until it reaches the spinal cord. So those are examples of long or short distances the signals travel. A response time endocrine system is where the hormones may take seconds to minutes to hours to days. So it can be fast or slow for endocrine glands, whereas for the nervous system, it's typically very, very quick. Action potential, as you know, is generated in milliseconds, and that triggers the release of neurotransmitters that are already there within the synaptic knob. So it's the quick release and therefore a quick response. All right, with this, let's look at the major endocrine glands. We will be talking about the hypothalamus, the pituitary, thyroid, adrenal glands, pancreas, ovaries, and testes. That's the main coverage. Major endocrine glands is how you should consider these. The others, there are a number of others actually. If you think about kidneys can make hormones, but that's not their main function. They have a much bigger role in the filtration of blood and production of urine, etc. So we will not talk about those types of situations here. Okay, some glands are strictly endocrine. Think about thyroid or adrenal. They have no other role but to make the hormones. Okay, similarly anti-pituitary. So they have this hormone production as their key function. Some have a major endocrine function along with some other major functions. Hypothalamus is a good example here. Hypothalamus makes important hormones. That's a major function, but it also has other major functions, such as the regulation of the autonomic nervous system, or body temperature regulation, the regulation of appetite, or satiety, things like that is where hypothalamus assumes many, many roles. And similarly, pancreas has two major roles. One is the digestive function, the other one is the endocrine function. Both are very important. So we would talk about pancreas here as well as you would talk about it along with the digestive functions. Okay? Okay, this time we'll do the classification of hormones. Okay, these are chemical substances. They're organized into 3 groups based on their chemical nature. Okay, the main group, the biggest group, is the group of peptide, polypeptide, and protein hormones. Okay, then you have the steroid hormones and the amine hormones. Now, you know the definition of peptide, polypeptide, and protein hormones. Peptide is a short chain of amino acids. Polypeptide is a long chain of amino acids, and protein is much longer chain of amino acids, correct? Protein hormones would probably have the three-dimensional configuration as well. Now, that's the main group. We will look at the examples in the next slide, but here is one that is shown, that's oxytocin, a peptide hormone. You can tell that these are the amino acids that are strung together. Okay. Next is steroid hormones. These are lipid hormones. Steroid is a type of lipid, and the steroid molecule is 4-ring structure. Note here, these are the 4 rings. One little modification could make this become one hormone or another. So you have 2 examples shown here, testosterone and progesterone. We'll look at other examples. So then the other group is amine hormones. Well, this stands for amino acid serving as a hormone. Amino acid tyrosine is the main one that serves as a hormone. Well, there is another amino acid, tryptophan, that could also serve as a hormone. We'll look at those examples very shortly. Here is one example shown. This is one amino acid. Now, a slight modification of tyrosine here would make it become norepinephrine. All right, here are the protein polypeptide peptide type of hormones. All the structures that make the protein peptide polypeptide type of hormones and all the hormones that are made from these tissues is all listed here. As you can tell, it's a long list, and we'll go through these at one point or another so we can stay tuned. Okay, steroid hormones, similarly, the tissue that secrete these hormones, and then the hormones themselves are shown here. Okay, so now amine hormones, similarly, it's a short list. Notice this is where I would say dopamine, T3 and T4, norepinephrine and epinephrine, all these come from tyrosine. Melatonin is the one that's from tryptophan. Okay, so that's the difference. Okay, with this, we will move on to talk about the mechanisms by which hormones bring about their effects at target cells. Well, now, you already know that there are 2 types of hormones. One group is water soluble, the other one is lipid soluble, or not water soluble, correct? Now, those that are water soluble hormones would need their receptors on the cell surface, not inside the cell, but on the cell membrane, facing the outside of the cell membrane. Why? Because the size would be bigger for these molecules to go through the membrane to find their receptors. Okay, so now what about lipid-soluble hormones? They can go through the membrane readily. Why? Because the membrane is mostly made of lipid, and these are lipid-soluble hormones, so they can move through the membrane and find their receptors inside the cell. Okay, so in here, we will look at the step-by-step events that are involved in the lipid-soluble hormone at its target cell. So the hormone here that's traveling along the blood finds its receptor inside the cell, and so it escapes and now binds to the receptor. Well, we could have added that it had a carrier protein. Now it separates from the carrier protein, moves out of the blood, and then that's not shown here. So you have to assume, because this is a lipid-soluble hormone, it should be carried in the blood by the carrier proteins. Okay, so assuming that, we will talk about the receptor location. It's an intracellular receptor because it's a lipid-soluble hormone. Now, intracellular meaning it could be in the cytoplasm or in the nucleus. Okay, so hormone binds to the receptor. This is when you call it as receptor hormone complex. The entire structure begins to move in the direction of the nucleus. Once it enters the nucleus, it binds to a specific location on the DNA. specific location on the DNA. Now, how does this complex know where to bind to within the DNA is a mystery, okay? Well, there are chemical signals, there are ways it's all controlled, but for us now, it is like it binds to one specific part of the DNA, okay? Receptor hormone complex reaches the DNA, binds to an exact location on the DNA. And once it binds to the DNA, that particular part of the DNA uncoils, and then the two strands separate. Two strands of DNA separate. One strand serves as a template and dictates its code, the triplet code that would be copied in the form of RNA. Okay, so this is what you call as transcription, where the DNA gives away its nucleotide-based sequence to be copied in the form of RNA. Okay, so thus the resulting RNA here is messenger RNA. It's carrying the message that's hidden in the DNA. Okay, this process is called genetic transcription. This is the first step in the synthesis of protein. Okay, so what have we learned so far? Let's step back. Hormone reaches the target cell, finds its receptor inside the cell. The complex of receptor and hormone travels in the direction of the DNA, binds to the DNA, uncoiling, and then the separation of the DNA strands and the copying of the bases, and then the construction of new RNA, that is messenger RNA. Okay, so that is genetic transcription, and that's how far we have gotten. The next thing is the messenger RNA that was just constructed would now travel out of the nucleus into the cytoplasm. Okay, so it's in the cytoplasm. What it has is the bases in a set of 3. You call those as codons. Okay, this is the complementary base pairing of the DNA. Okay, if DNA has ACC, it would be copied as UGG, for example. So that is codon language that you see on the messenger RNA. Well, soon what happens is ribosomes that are nearby would come and attach to this RNA. Ribosomes have 2 subunits, large and small. They separate and then hold on to the messenger RNA, and the 2 subunits come together. That's how messenger RNA is now trapped by the ribosomes. Okay, well, once this is going on, you also have tRNA. tRNA structure is where you have amino acid on one end, and the 3 base sequence, you call it as anticodon, that's on the other side. So you have tRNA that arrives towards the messenger RNA that's in the cytoplasm. And if the codon and anticodon match, then the tRNA would leave the amino acid that it carried in that place, and then a new tRNA would bring a new amino acid, and so on. That is how a protein is constructed using this messenger RNA and the codon sequence that's present here. And this process is what you call as translation. Okay, transcription and translation are the 2 steps in a protein synthesis. So what we see here is a response that is given by the cell in response to a hormone. The hormone arrived, and what all happened here? A long series of steps took place, and the end result is the production of one protein. This protein could be an enzyme, could be an antibody, could be another hormone. We are not looking at what that is. What we are saying is a hormone is a messenger, and it would make the cell respond to it and do something within the cell. That something happens to be the production of this new protein in this example. Okay, now we will turn around and look at a different type of scenario. That is, you have a water-soluble hormone, and it has to have receptors on the cell membrane. Okay, cell membrane. Now, we already know why, because the hormone is too big to make its way into the cell, because the cell membrane is all lipid-based, and the water-soluble hormone is typically amino acid or a peptide, polypeptide, protein type of hormone. Okay, having understood that, let's look at what's going on here. You're talking about a receptor on the cell membrane, as expected, correct? So here is the example of a water-soluble hormone traveling in the blood, and it finds its receptor on its target cell. It leaves the blood and reaches the receptor and binds to it. Okay, so that's the first step, binding of the hormone to the receptor. Okay, the membrane receptor that you see here is often associated with a G protein. It's always there, but when the hormone binds to the receptor, G protein gets activated. Activated G protein would lead to a series of changes within the cell. Okay, one set of changes is addressed here. We can look at another set of changes in another place. Okay, so what have we learned so far? That is the binding of the hormone to the receptor, activation of G protein, and that then activates an enzyme. How does this happen? Well, you have 3 subunits in the G protein structure: alpha, beta, gamma. Okay, alpha subunit separates from the beta gamma subunit. Beta gamma subunit does not purge, but the alpha subunit is the one that moves and then binds to adenylyl cyclase. This is the enzyme. Now this is activated. Again, it's a series of events that happen soon after hormone binds to its receptor. We have now covered 3 steps here. One is hormone binding to the receptor, 2, G protein activation, 3, adenylyl cyclase activation. Adenylyl cyclase is also called as adenylate cyclase, by the way. Okay, this enzyme is the one that can break ATP and release cyclic AMP. Okay, so this phosphorylation reaction occurs, and you suddenly have a large amount of cyclic AMP within the cell. Okay, until the hormone arrived, there was no significant amount of cyclic AMP, and so the situation was of one type. But in the presence of the hormone, you have high amount of cyclic AMP. That itself is a trigger for the cell to turn on another set of events. Okay, what does cyclic AMP do here? Well, note here, it activates one set of proteins called protein kinases, protein kinase A. Okay, so protein kinase A is activated, and now that would lead to yet another series of events. All right, so that is what we call as a response to the hormone. So let's review the steps one more time. Hormone binds to its receptor. This is a membrane receptor. G protein activation. Then the adenylyl cyclase activation, conversion of ATP to cyclic AMP, increased levels of cyclic AMP, activation of one set of enzymes called protein kinases, or protein kinase A to be more specific, which then turns on many more cellular events. What those cellular events varies from one hormone to another and one cell to another. So we're not going to spell out all those specific details, but this is a global effect of the hormone at the target cell. Okay? What we're looking at here is the alternate set of events by G protein activation. Okay, so here you're looking at the membrane receptor again. That means these are water-soluble hormones. Okay, water-soluble hormone activating cell membrane receptors, binding to cell membrane receptor, G protein activation. Now, G protein activation, this is what happens. There is a phospholipid in the cell membrane. It is now targeted by the activated G protein. Okay, by having phospholipase C activation. Okay, so in the presence of G protein activation, phospholipase C is activated. Okay, which then would turn on one of these two sets of events. Phospholipase C could convert PIP2, this is a phospholipid, to IP3, that is inositol triphosphate. Okay, inositol triphosphate basically releases endogenous calcium sources. So it releases calcium from whatever source we have. Endoplasmic reticulum, smooth endoplasmic reticulum typically stores calcium ion. So that is released, and now calcium ion is the messenger within the cell. It would now bind to comardulin. Comardulin is a protein that is activated, and now what happens is it would turn on muscle contraction mechanism. Okay, it's typically seen in a smooth muscle. When smooth muscle has to be contracting, this is the type of mechanism you see. What happens is G protein activation activates phospholipase C. Phospholipase C converts PIP2 to IP3, or DAG is diacylglycerol. Okay, so now IP3 would release calcium ion. Calcium ion binds to comardulin, activates comardulin, and that would trigger muscle contraction. Okay, smooth muscle contraction is what you would expect. Or it could be the muscle that is in the blood vessel lining. If a blood vessel, smooth muscle is contracting, you may be seeing vasoconstriction in response to that. That's how, this is exactly how vasopressin, or antidiuretic hormone, brings about its effect, you see. Or angiotensin 2 also brings about its effect in this fashion. Okay, so the next one is phospholipase C converting PIP2 to diacylglycerol. What happens here? Activation of a new set of hormones, protein kinase C activation. So that then leads to a new set of events like phosphorylation of various substrates and the new end products. So let's review this again. What we're saying here is that hormone binding to a membrane receptor can turn on G protein mechanism. G protein mechanism could be of one sort or another. So we looked at two examples. The first one we looked at is the activation of adenylyl cyclase, increased levels of cyclic AMP. Cyclic AMP is often referred to as the second messenger. Why? Because the first messenger, the hormone, is outside of the cell, is not able to arrive inside to trigger changes within the cell. So it is cyclic AMP that turns on the events within the cell, and therefore it's the second messenger. So now the second mechanism that we looked at is the phospholipid being targeted by phospholipase C, G protein activation of phospholipase C. It converts PIP2 to IP3 or DAG. IP3 would trigger muscle contraction by activating comardulin. Diacylglycerol could turn on one or another set of events within the cell, just like a protein kinase A activation that we saw before. Okay, so here calcium ion could be a second messenger. All right, so those are some ways of referring to how other agents could take over the function of the hormone. But always remember that without the first messenger, there is no second messenger. Hormone has to be there to turn on these set of changes. Okay, now we look at some other details. How is hormone cleared from the body? Okay, this is where we need to think about one important detail. The hormone that's made is not going to be around forever. Okay, its effect is brought about at the target cell, and soon we need to clear it. We need to clear this, otherwise you may end up obtaining undesired effects. You do not want this hormone to be lingering around forever. How do we clear? Well, if the membrane receptor is inside the cell, then the hormone is already inside the cell, and the intercellular mechanisms would break it down. So the hormone is not around anymore. Metabolic breakdown of the hormone within the target cells. And if it's a water-soluble hormone, remember how water-soluble hormone is not able to enter the cell. That's where you would have the enzymatic breakdown of a hormone in the blood or at the tissue level. And then liver breaks down hormones as well. Some hormones are taken to the liver where it processes and is inactivated and removed in the form of bile. And kidneys do the same. Some hormones are processed in the kidneys, and any metabolic wastes are removed in the form of urine. Okay, good. Here is a nice summary of the hormones that are bound versus the hormones that are not bound, and then the half-life of these hormones and the clearance time. You don't have to memorize this. This is just a good reference, but you know already that thyroid hormones and steroid hormones require carrier proteins. Protein binding is therefore much higher. Now, protein hormones do not require carrier proteins. Now, if you look at the carrier protein bound hormones, the half-life is days, 6 days, 1 day, or hours, or a longer period of time, like 25 minutes, 85 minutes. Whereas protein hormones could be degraded sooner. 8 minutes is how quickly some of them are inactivated. We have angiotensin 2. That's its half-life is only like 1 or 2 minutes. Okay, so that's how interesting these things are. Clearance time is given. Some require a long period of time, whereas some can be cleared right away. Okay. Now some definitions is what we are talking about. Permissive effect. In some cases, the presence of a hormone is essential for the effectiveness of the other hormone. Okay, so that is where you have that permissive effect. All right, in the presence of estrogen, progesterone receptors are added, and this type of relationship is what you're talking about. Presence of one hormone basically enhances the ability of the other hormone. Okay, so when you think about a set of hormones, LH and FSH, there may be that interrelationship that exists. You see, synergistic effect. Two hormones with similar effects produce better response. If you think about hormones that increase blood glucose levels, glucagon and cortisol, what do you call that? That would be a synergistic effect, because both are working towards increasing blood glucose levels. Antagonistic effect is when the hormones act in the opposite ways, two hormones having opposing effects. Okay, you could think about insulin and glucagon as a good example of how hormones could be quite opposite to one another. Okay, insulin reduces the amount of glucose that's in the blood by stimulating the uptake of glucose by the cells, whereas glucagon increases blood glucose levels by breaking down the sources of glucose within the cells. All right. Now let's talk about what regulates hormone secretion. Okay, is it the nervous system that controls the level of hormone to be around, or what other mechanisms exist? That's where we talk about a couple of different ways of regulating hormone secretion. One, of course, is the nervous system. Not all hormones are regulated by the nervous system, however. Okay? A growth hormone is probably the nervous system regulation. You know why? A growth hormone releasing hormone is produced by the hypothalamus. You see, stress and other stimuli would make you produce growth hormone, releasing hormone, that in turn triggers the production of growth hormone, that in turn releases, make you produce more muscle and increase the length of the bone, etc. And that's how you create growth. So that's one example. Or cortisol that's released in response to stress. That's, again, the nervous system regulation. Now, what about other hormones? What about insulin, for example? Does the nervous system control the secretion of insulin? No. What controls the secretion of insulin is the amount of glucose that's present in the blood, you see? So these are solutes that control the production of certain hormones. Solutes like glucose or calcium ion may dictate the presence or absence of certain hormones. And however it is, one thing you need to know is there is what we call as feedback mechanism. If you have a hormone in a lesser amounts, you can send negative feedback signal and make the gland produce more hormone. Okay, so that's how you control the levels of hormones, through negative feedback mechanism. So that's what is shown here. Okay, so you have this signal here. Homeostasis is when everything is normal. Okay, but when there is an imbalance, you need more hormone to be released. Okay, signal is taken to the hypothalamus, and in turn you would release the hormone, and then you would maintain homeostasis. Got it? So negative feedback mechanism dominates in the regulation of hormones. However, positive feedback mechanism is essential in some rare situations. Two of those are one with ovulation. Okay, in order for ovulation to occur, you need a surge of several hormones, estrogen, LH, FSH, and that's where you have the positive feedback mechanism that is initiated at some point. Then another positive feedback mechanism is true during the induction of labor. Oxytocin release is regulated in a positive feedback manner until childbirth, labor happens. Okay, so that's feedback mechanism and how hormone secretion is controlled. All right, primary and secondary secretion. Okay, what that means here is if a gland makes a hormone and it controls the target structure, that means it's primary. Okay, this is the hormone that's made and it's acting at the target gland. And if you need that hormone in larger amounts, you have negative feedback. So that is how you control. Hyposecretion is undersecretion. That's when you make the gland release more and affect the target. So if there is a primary effect, if this gland is not functioning, then this target gland function is lost, and that is what you call as primary hypofunction. Correct? Now, let's talk about yet another mechanism. That is this one here. This is the gland, okay? It makes this substance shown in blue. It acts on the target gland, which in turn affects the release of yet another set of hormones. So here, this target gland is controlled by this particular endocrine gland. So this is one level of control. And this gland now controls a target tissue. You see? So if this gland is abnormal, you would affect this target tissue. If this gland is abnormal, you would affect this target tissue. But this is the most direct, correct? So that would be primary effect. This would be secondary effect. Okay, so everything is normal here, but this gland is not able to release this substance, this particular hormone, which in turn would be able to produce this particular hormone to control the target tissue function. So that's when you would talk about this as secondary versus primary. Okay, good. Now, there are situations where you need more receptors to respond to the hormone, and that's what you call as upregulation. More receptors are added as needed, and or if it's not the number, it may be the sensitivity of the receptor that would be altered. Okay, that is what you call as upregulation. So if you have a hormone that is important in the accomplishment of a certain function, you may want to add more receptors quickly. And this is what happens in the induction of labor. You have oxytocin, but you just add a large number of oxytocin receptors in the smooth muscle lining of the uterus towards the very end of the pregnancy. All right, down regulation is the opposite. Okay, you have a hormone that's around, but that function is not desired anymore. So how do you control it? Instead of having the same number of receptors, you would remove the receptors or remove the sensitivity of the receptors, and that's how you would downregulate the action of a particular hormone. And a missing element here that is important for you to know is polypeptide and protein hormones can be made ahead of time and are stored in their secretory vesicles. Insulin, for example, is stored there in large amounts. Only when you need it, it's immediately released. So that's possible with polypeptide and protein hormones, but not with steroid hormones. Steroid hormones have to be made as they're needed, and their starting point is arachidonic acid. Arachidonic acid is a lipid molecule that is converted into a steroid hormone as needed, okay, and is not stored. Good. With this, we will close this lecture and talk about the new topic later. Thank you.